How the construction site of long-term memory gets built in the brain. How the construction site of long-term memory gets built in the brain. You're listening to Technical News Reading, presented by Hakeem Alibokis Alexander on Spreaker, social podcasting, presented for World Reading Club in association with Uniquilibrium. This edition's reading focus comes to us from neurosciencenews.com and is titled How the Construction Site of Long-Term Memory Gets Built in the Brain. It was published on February 6, 2023 under the category of Neuroscience. Summary. Study identifies a set of proteins that undergo experience-dependent changes while forming both short-term and long-term memories. Source: University of Florida. Think of a new, longer-term memory as a construction site inside the brain. The brain's neurons restructure themselves and build or demolish connections with other neurons to store the memory for retrieval when needed. The neurons don't do the work without help. They need building materials from a distant warehouse, so trucks hit the highway to transport cargo to the construction site. The cargo of those trucks varies over time depending on the strength of the memory. Do the neurons need supplies to build a structure that endures hours, days, weeks, or even years? Researchers at the Herbert Wortham UF Scripps Institute for Biomedical Innovation and Technology have discovered that these cellular building materials, in this case, sets of proteins, undergo experience-dependent changes while forming short and long-term memories. A paper on the discovery by a team of Wortham UF Scripps Institute scientists was published on January 5th in eNeuro, an open-access journal of the Society for Neuroscience. It's a glimpse into the brain's plasticity, or its ability to adapt and change its structure as we live our lives and accumulate memories. Additionally, it enlightens future research about how the brain's enormously complex systems operate. That has potential implications, scientists said, for better understanding neurological disorders. Those include Alzheimer's and amyotrophic lateral sclerosis, known as ALS or Lou Gehrig's disease. This is giving us a much more detailed insight into a process that we know is important for memory, said senior author Satya Narayanan V. Putan Vital, Ph.D. He is an associate professor at the Wortham UF Scripps Institute. The connections of these neurons need to be selectively modified to form long-term memory, he added. And for the modification to occur, the neuron needs to send materials from the cell's soma to its distant synapses. Unique packets of proteins are sent, and this cargo of proteins changes over time as memory is encoded. This is an exciting finding. The soma is the main cell body of a neuron, where its nucleus resides, while synapses are the place where connections between neurons are built. On a cellular level, it's a long trip. Putan Vital's team used a species of sea slug called Aplasia to explore how memories are encoded. The slugs have gigantic neurons compared with those in the human brain, making them easier to study. A neural mechanism for storing certain memories in slugs is thought to be extremely similar to what occurs in the human brain, Putan Vital said. Memory, of course, is the essence of what it means to be human. Sometimes memories are short-lived, as when we see a stranger's face at a party and cannot recall it the next day. A simple biochemical change in the brain creates shorter-term memories that are with us for several minutes to a few hours, he said. Here we have a stained image, probably of one of the neurons, which caption reads, "A species of sea slug called Aplasia bottom squirts a purple ink to ward off predators in this close-up view by Wortham UF Scripps Institute scientists. Researchers used a sea slug to examine how neurons store memory. The slug's neurons are gigantic compared with humans, making them easier to study. The credit goes to Abhishek Sadhu." Neuroscience News continues. Longer-term memories, however, bring out the full construction crew that indelibly encodes the brain's circuitry, especially when the brain is sensitized to a strong event, a car crash, a child's birth, the moment someone learns shocking news. Putin Vital said how the brain accomplishes this has been poorly understood, and even now, it will take much more research to fully decipher the cellular building blocks that form memory. The material that moves between soma and synapse includes numerous proteins, which are crucial workhorses that drive many of the processes and chemical reactions that allow human cells to function and carry out tasks. Other cellular materials might also change over time, Putin Vital said, but that will be a topic of future research. About this memory and learning research news. Author Eric Hamilton, source University of Florida. Contact Eric Hamilton, University of Florida. The image previously mentioned in the article on Neuroscience News is in the public domain and is credited to Abhishek Sadhu. The original research is under open access and is from a paper titled "Short-Term and Long-Term Sensitization Differentially Alters the Composition of an Anterograde Transport Complex in Aplasia" by Satya Rayanan, Putin Vital, and others in eNeuro. You've been listening to Technical News Reading, presented by Hakeem Alibokis Alexander on Spreaker, social podcasting, presented for World Reading Club in association with Uniquilibrium. NeuroscienceNews.com continues with an abstract from the paper titled "Short-Term and Long-Term Sensitization Differentially Alters the Composition of an Anterograde Transport Complex in Aplasia." Long-term memory formation requires anterograde transport of protein from the soma of a neuron to its distal synaptic terminals. This allows new synaptic connections to be grown and existing ones remodeled. However, we do not yet know which proteins are transported to synapses in response to activity and temporal regulation. Here, using quantitative mass spectrometry, we have profiled anterograde protein cargoes of a learning-regulated molecular motor protein kinesin, Aplasia kinesin heavy chain 1, or APKHC1, following short-term sensitization, STS, and long-term sensitization, LTS, in Aplasia californica. Our results reveal enrichment of specific proteins associated with APKHC1 following both short-term synchronization or sensitization and long-term sensitization, STS and LTS respectively, as well as temporal changes within 1 and 3 hours of LTS training. A significant number of proteins enriched in the APKHC1 complex participate in synaptic function, and, while some are ubiquitously enriched across training conditions, a few are enriched in response to specific training. For instance, factors aiding new synapse formation, such as synaptogmin 1, dynamin 1, and calmodulin are differently enriched in anterograde complexes 1 hour after LTS, but are depleted 3 hours after LTS. Proteins including gelsolin-like protein 2 and Sec23A, Sec24A, which function in actin filament stabilization and vesicle transport, respectively, are enriched in cargos 3 hours after LTS. These results establish that the composition of anterograde transport complexes undergo experience-dependent specific changes and illuminate dynamic changes in the communication between soma and synapse during learning. NeuroscienceNews.com asks that you join our newsletter. Sign up to receive our recent neuroscience headlines and summaries sent to your email once a day, totally free. We hate spam and only use your email to contact you about newsletters. You can cancel your subscription anytime. Neuroscience News posts science research news from labs, universities, hospitals, and news departments around the world. Science articles can cover neuroscience, psychology, AI, robotics, neurology, brain cancer, mental health, machine learning, autism, Parkinson's, Alzheimer's, brain research, depression, and other topics related to cognitive sciences. You've been listening to Technical News Reading, presented by Hakeem Alibokas Alexander on Spreaker, social podcasting, presented for World Reading Club in association with Uniquilibrium. This edition's reading focus comes to us from neurosciencenews.com and is titled How the Construction Site of Long-Term Memory Gets Built in the Brain. It was published on February 6, 2023 under the category of neuroscience. You can read this article and others like it for yourself by visiting neurosciencenews.com. That's neurosciencenews.com. Neurosciencenews.com. How the construction site of long-term memory gets built in the brain.